Hello friends, in this session we will going to see the how to do the path analysis in Excel. This is very important topic. So what exactly you will get in this Excel sheet we will going to discuss in this session. Okay, we continue this session so you will get the clear cut idea about the how to do the path analysis in Excel with the diagrams. Okay, so for example, you can see here we have the data for the example. You can see here the x1, x2, x3 and y. You can consider this is the last parameter as a dependent parameter. Okay, this is dependent on the other character. You can consider x1 as a plant height, then x2 as a number of branches, then you can consider x3 as a number of seeds or any character or parameter as per your research. We will develop this type of Excel sheets for the data analysis and we will sell this type of Excel sheet as well. If you need this type of Excel sheet, then definitely contact us. You will get my WhatsApp number in the description of this video, so that you just go to the description of this video and contact me, so that you will get the this type of Excel sheet. But remember, this is the paid Excel sheet. Okay, so we continue with us in this session, so you will get the clear cut idea about what exactly you will get in this session or in this Excel sheet. Okay, so let me zoom it for you. You can see here everything, but it's not, I think, so visible, so that I will just zoom it. Okay, at the end we will going to see the how to draw the path diagrams. Okay, so be continue with this session. Okay, so don't left between this. So I'll zoom it. Okay, now I think so it's visible to everyone. You can see here the path analysis. You can see first here you should have to write here the character name. Whatever a character you choose for the data analysis, you can put here instead of x1. Then same for the x2, x3 and yield. Yield is our dependent. You can see here the dependent character. You can see character, or you can call it just yield, or you can call it a parameter. Okay, all terms are equal. So you can see here what exactly you want to do in this Excel sheet. It's very simple because this is a fully automatic Excel sheet. So you can just go to your data. For example, you have the R1, R2, R3 with respective treatments. You can see here T1 up to T12 treatments, and this is data for R1, R2, R3. You need to just select your data, then copy, just come back to our design. You can see here the x1 character. In that you see here the treatments or genotypes. You see here the 12 treatments, then 3 replications. You see this is the corresponding value. Okay, so select first cell, then right click, then you can choose here the paste as a values. Don't paste like a control C and control V like that. Don't do that. Okay, just you come here and you see here the paste values. Okay, just click on it. You'll get whole data over here, and this is fully automatic, so you don't need to do anything. You just copy paste, or just you can manually enter your data in each cell with respective of the treatments and replication. Okay, rest of the things it will do. Okay, so we will explain what exactly you will get from this Excel sheet. Okay, so first you can see here you will get the ANOVA tables. Okay, you can see here the x1 is our the first character, right? So that for first character you will get the ANOVA table. ANOVA, it means the analysis of variance table. In that you will get the variation, bifurcation, like replications, treatment error and total. Okay, so you can see here you will get the significant values 5% as well as 1%, and you see here the values as a significance, non-significant or significant. Okay, if SIG, that is significant, NS non-significant. Okay, and in that you also get CV value. It's very important, that is coefficient of variation values you will get here. Also you will get the standard error mean, then standard error difference, then CD for 5% and CD for 1%. Okay, this is for the character 1 or character first or parameter first. Okay, so second also the similar things, third also similar, and last over the dependent parameter also the similar thing, because the rest of the all parameters are independent. Okay, and yield are the dependent parameter. So below that you will get the ANOCOVA tables. ANOCOVA, it means the analysis of covariance tables. It's a combination of two parameters. We are chasing each other so that we will get the combination of that particular parameter. Okay, for example, x1 versus x2, then x1 versus x3, then x1 versus yield parameter, that is the dependent parameter, then another x2 versus x3. Okay, likewise we are pairing these parameters, and we will developing the ANOCOVA. ANOCOVA also you will get the significant for the 1% as well as the 5%. Okay, you can see here the sum of square, mean sum of square, everything you will get in this design, and this is the fully automatic design, so that you don't do anything over here or don't change any formula. Just you need to enter your data or just copy paste your data as values. Okay, so below that this is the our calculation part. This is not needed for you. We will explain in the next what exactly you need to put in your thesis. Okay, this is for a rough work, so that you will get the phenotypic correlation coefficient, genotypic and environmental. If you want this type of correlation table, you can copy paste this table. Okay, this is the fully matrix table. Okay, so next is very important. So what exactly you want to put in your thesis? Okay, so you can see here you need to put this type of min table in your final thesis tables. In that you can see here the genotypes and treatments. You see here the x1, x2, x3 and yield. Yield is our dependent parameter, and x1, x2, x3 are our independent parameters. Okay, so in that you will get the overall means, that is we called as the grand mean. You can see here the grand mean for x1, x2, x3 and y. You need to just copy paste this table and you format as per your thesis or university guidelines, and you can make that type. Okay, so you will get here the standard error difference. You can put here the standard error mean as well, because all means and differences already given over here. Okay, you can see here the individual ANOVA table, you will get everything. Okay, if you want to put the CV, you can take CV from this. If you want to put CD, then you can take a CD for 5% as well as the 1% if you need. If you want to put a standard error mean, you can put the standard error mean, otherwise you can put the standard error difference. Okay, you can see here everything you will get in the final treatment mean tables. Okay, below that it's very important, and many softwares and online softwares, they don't provide the everything in a single sheet, so that we will try to provide everything needed for your thesis in a single sheet, and we arranged all the parameters and all the needed thesis tables in a single Excel sheet, so that you will get everything as analyzed and informated as per your thesis. Okay, so in genetics parameter, you can see here the genetics parameter or genetic parameter. Okay, so in that you should have to consider the parameters like x1, x2, x3 and yield parameter. In that you will get the maximum values for the x1, x2, x3 and y, then minimum value. Then it's very important, you will get the environmental variance, then genotypic, phenotypic, GVPV, that is the genotypic variance, phenotypic variance. And many times this is the environmental coefficient of variation or environmental coefficient of variance, ECV, GCV and PCV. Okay, here you can see the heritability, that is H square or H hat 2 we call it. It's having two types. Okay, first is the broad sense and second is the narrow sense. We consider in this example broad sense. If you want narrow sense, then we will add that also, because we develop this type of Excel sheets for the students as per their need. So if anybody want this type of Excel sheet, then definitely contact me. My contact number or WhatsApp number I already mentioned in the description box of this video, so that just go to the description box of this video and you will get the my contact number and just click on the link and just contact me on WhatsApp. I'll give the pricing for this because this is a paid Excel sheet. Okay, and if you want the analysis, we will provide the all type of analysis services as well, so that you can contact us if you need. Okay, so next you will get the genetic advance, that is GA, for 1%, 5% and 10% as per the your outline of research work or synopsis you can consider. Okay, then next genetic advance as a percentage over the mean, that is GAM or EGA, both are the equal. So 1%, 5% and 10%, and last you will get the range, right? So that maximum, minimum, and you will get the range over here. This is our the second table for the thesis. Then next you will get the variance. You can see here the values for the variance. Variance as a diagonal and covariance as a off diagonal. Okay, so you can consider here the variance and covariance table. This is the diagonal values. Okay, these are the variance and these the rest of the values are the covariance. You can consider here. Okay, this first one, second, third and fourth, these four values are diagonal values, that is the variance values, and rest of the all values are off diagonal, that is the covariance values in this matrix. Okay, you can consider here the first character x1, x2, x3 and your yield, x1, x2, x3 and yield. Okay, it's very important right now, because above topics are the genetic parameters also included. Okay, but most interesting and important are the correlation analysis. Okay, because we considering the path analysis, so that we bifurcating that the correlation coefficient in the different types. Okay, so that you can see here the correlation analysis, in that you will get the phenotypic correlation coefficient. It's very important. Okay, if you want without non-significant, significant for the 1% and 5%, then you can consider this last table. Already I explained this table. Okay, this is the phenotypic correlation coefficient table. You can consider this table. Okay, because this is the final thesis table, so that we put everything which is needed, that is non-significant, significant for 1% and 5%. You can see here the stars as well. Okay, this is the 2 stars. You can see here the 1% and 5%. 1% you will get the 2 stars and 5% you will get the single star. Okay, remember this. And if your character or phenotypic correlation coefficient is non-significant, then you will get the non-significant as a NS. Okay, so next is the genotypic correlation coefficient. This is the same like a phenotypic. You can see here the non-significant, here is a 1 star, here is a 2 star. 1 star it means 5% and 2 star it means 1%, and NS it means non-significant. Okay, the rest of the environmental correlation coefficient. In that you will get the environmental values for the correlation coefficient, and below that you will get the correlated response as well, how it's correlated, that response you will get here. Okay, we consider, keep in mind, we consider the yield as a dependent parameter. Okay, there is a different types, so we can consider the two dependent parameters or a single yield as a dependent parameter. Likewise, we can develop this type of Excel sheet. If you want, then definitely contact us. It's a paid Excel sheet. If you want, then definitely contact us. Okay, then below you will get the path analysis. Okay, so this is the correlation coefficients and this is the path analysis. We are now reaching at our main goal, that is the path analysis. Okay, so in this table you will get the genotypic path coefficients as a direct, that is diagonal, and indirect effect is a off diagonal. You can see here the I already highlighted with the bold letters or bold numericals, so that you can see here this is the diagonal data. Okay, so this is the for genotypic path coefficients, and these diagonals are the direct effect. Okay, you can see here the only x1, x2 and x3. Why it's x1, x2 and x3? Because we consider the y versus x1, y versus, because we consider y as a dependent parameter. Okay, so that we consider here the y versus x1, y versus x2, y versus x3, and you will get the residual values as well. So here is the direct effect and here is the indirect effect, and you can see here this is the genotypic, this is the genotypic path coefficients. Okay, this is the aggregate of this all. You can see here the aggregates. Okay, sum is minus 2.21. okay, you can see here this is the sum. Okay, so next you will get the phenotypic path coefficient, same like the genotypic. You can see here in diagonal you will get the direct effect, and in off diagonal you will get the indirect effect, and also you will get the residual value. Okay, so next environmental path coefficients, same like above, diagonal as a direct and off diagonal as a indirect effect. Okay, so this is all tables are very important for your thesis. Okay, we included all the tables which is needed for your research. Okay, everything we included. Also we included 1%, 2%, then 1 star, 2 star, non-significant, significant, everything included in this Excel sheet. Okay, so it's a very important topic because many students struggle to draw the path diagrams. Okay, it's very easy because already we set up that path diagrams in Excel sheet, so you will get the two different types of path diagrams. Okay, because we developed this easy path diagram in the Excel sheet, so you will get the clear cut idea about this. So you can see here genotypic path diagram, you can put here the yield, then you should have to consider your residual effect. You can see here your residual effect. Okay, already we discussed on that. You can see here residual, right? So you can put that residual in our diagrams. You can see here, if you want this type of diagrams, we also provide this type of diagrams. Okay, so in PNG or JPG format, because in this Excel you will get just the format of that diagram, but as per your need we will develop that diagram in software, so that if you need this type of diagram, then definitely contact us. You will get my contact number in the description box of this video, so that you just go to the description box of this video and contact me. Okay, all services are paid services. Okay, so next you can see here you will get the x1, x2, x3, and these are the direct effects on the yield, and these are the indirect effects on the yield. Okay, so this is the same data but the different diagram structure. So you can see here the genotypic path diagram. You can see here all things. You will get this also in this Excel sheet. You don't need to do anything, just you need to put the R1, R2, R3 with respect of the your treatments values, and you will get everything, because this is the fully automatic sheet. Then same for the phenotypic path diagrams. Okay, for phenotype you will get the same diagrams, and for environmental also you will get the same diagrams. I just put that two example diagrams format over here. If you want the different format, then definitely contact us, then we will provide you. Okay, and this is all about the path analysis in Excel. It's a very simple, easy, because you need to just put this the red color values and nothing else, and you will get the everything, and you will get also the final thesis tables. Okay, and just you copy paste wherever you want and you complete your thesis. Okay, if you need any help regarding this, then definitely contact us. You'll get my contact number in the description box of this video, and just hit the link and message me over the WhatsApp, and we will discuss on that. But you remember one thing, all services are paid services regarding the data analysis, data manipulation and other things. Okay, so that you definitely contact us, and we sell this type of Excel sheet. If you want, then definitely contact us. Thank you so much.